Good evening, everyone. Hope I'm audible. Okay, so let's begin the biochemistry chapter, which is Unit 1. And I told you few things about Unit 1 in the last class, right? I told you which part to look for in Unit 1. And if it's possible, you can, if all of us, most of the time, we already uh, learned biochemistry at some part of our time. So if you learn it from the previous books like Leninger or Voigt and Voigt, any other book like that, you can follow that to recapitulate your idea. But if you are new to this field, I don't think much of people are new to the field of biochemistry here. But as biochemistry is a large unit, there will be a lot of things included in there, right? All the biochemical pathways will be there, and the protein structures, all the macromolecule structures are also there. So this is a very much versatile chapter to ask you questions from different domains. So in this case, I told you earlier and I'm telling you again, it's better that you prepare those things that you already know at some part of your study. If you're from biochemistry, molecular biology, microbiology, biotechnology background, you probably have already had an understanding of biochemistry, the basics understanding of biochemistry. So uh, read those part of the biochemistry along with some other books. Lippincotts Illustrated Review is very handy book in terms of this part, because our goal here is to learn things faster, to catch up with the things faster. So I'll recommend you to follow the Lippincotts Illustrated Review of Biochemistry, because that book is written with mind map, right? It is also written with the help of flowcharts and a lot of diagrams, which will really be helpful for you to grasp the idea. And along with that, we need to prepare a lot of mathematic part of the bioinformatics. And I told you there are different portions of biochemistry which will be really important for the mathematic aspects, and we'll solve that as well. But our sole target here will be, because if you look at a question of biochemistry, you'll find out that from this part, over 50% questions are mathematical, and the rest of the 40% questions are theoretical. So it's always a better idea to solve and practice those mathematical questions, because no matter how much you read the theory, you're only going to answer the 40% of the theory questions. Okay, so keep that thing in your mind. And today actually I'll be talking about some fragmented topics of biochemistry. And literally, as you see, two classes are allotted. So in reality, in two classes you can't even cover the introduction of biochemistry. So what we are going to talk about biochemistry here are the topics which I find are the most important regions of biochemistry to give questions from. Okay, so read this part, and apart from that, whatever other things that you need to prepare, I tell you the name of the topics, and it's better that you follow Lippincotts Illustrated Reviews. And if you forget most of the part from the biochemistry part, then you can go and read Leninger. Okay, so first things first, let's talk a little bit about the enzymes. And we need to have an important, interesting idea about the enzymes and how enzymes work, because all of us know the what is enzyme and what are their function. We know that enzymes are involved in what we can say simplifying any chemical reaction, right? So it's as a catalyst of any kind of chemical reaction that is going on inside the cell, right? So whatever thing we've learned so far, like helicase, it was an enzyme. DNA polymerase, so all these things, these are enzymes, and we know all these enzymes are proteins most of the part, and they involved in the process of all the other biological functions, cellular activities inside our body. So what we know about enzyme is, let's take this enzyme, and enzyme always interact with the substrate, and at the end what we want, we need the product. We need the product at the end, because our journey starts from substrate, ends in product, and at the end, along with product, we also recycle the enzyme, and that enzyme remains as it is at the beginning of the reaction and at the end of the reaction. So enzyme literally is not altered by its structure. It's not involved in any kind of uh, covalent like it's not involved in any kind of permanent structural change in it. Okay, but helps in conversion of substrate into the product. Now the question is, if we don't put the enzyme, if we just put the substrate, and now again the goal is to produce the product, can it be achieved? The answer is yes, it can be achieved. But for any chemical reaction to occur, what we need to cross the boundary of an energy barrier. Which energy barrier? It's known as the activation energy. So there is a conversion of substrate into the product. So this conversion will take place without the presence of an enzyme, but it will be slower conversion. And it will also the activation energy, which is like a hurdle to transfer between a structure of substrate to the product, that hurdle will be more. Okay, that hurdle is more. So the activation energy is more in case of without the enzyme. But whenever we are placing enzyme next to the substrate, that activation energy lowers down. So the activation energy becomes less. So the hurdle is now small. So substrate can be converted to the product pretty fast. That is the beauty of enzyme. Now the question is, what is in between? What's happening in there when an enzyme is linked to an substrate? That's helping to lower down the activation energy and reaching the product faster. That's the question. What is going on there and why is this happening? Because it's simply enzyme is not changed at the end of the reaction. It remains as it is. So definitely there is everything what's going on is going on with the substrate itself, right? But what this enzyme is doing, it's placing the substrate and all the reactants of a chemical reaction close to each other. That's what an enzyme can do. It brings them close to each other, because in reality the chemical reactions where we generally involved in making of a bond or breaking of a bond. So making and breaking of bonds can be done by different groups, different chemical groups that are present in different reactants. So enzyme is not doing any of that. Enzyme is not providing any of this kind of function. But what enzyme is doing is bringing the substrates close to each other. So let's say if there are two reactants, reactants 1 and reactants 2, and that produce a product. So both the reactants should be closely placed. So enzyme here forms a structure in between known as the enzyme substrate complex. And this is by far the most important structure that can be achieved by bringing the reactants close to each other, because once they form this enzyme substrate complex, then the conversion of this enzyme substrate complex to the product become easier. So there are these two steps of the reaction now, as you can see, when the first step is enzyme and substrate separate, and they can start association and forming enzyme substrate complex. Now this case there can be association or dissociation, but both of these things are really like this association and dissociation of enzyme substrate complex is faster. It's faster. Convert, like one enzyme is there, enzyme has their active site open, substrate can go there and bind to it. So once substrate binds to the active site of the enzyme, they have this eager affinity towards each other. So enzyme substrate complex formation is easy, it's fast. But the second stage of the reaction, that is a conversion of the substrate into the product and removal of the product, that is slow, because that take more time. That is the actual process of conversion of the substrate, modifying its structure into a product. So this clearance is taking more time. Association of enzyme and substrate takes less time. Okay, so now if we look at this particular scheme, enzyme substrate forming enzyme substrate complex and then convert it to the product, if we look at this scheme, we can assume something. What is it? If you find this enzyme substrate complex as a component, let's say there are three separate components. One is the enzyme and substrate separately. Another one is the enzyme substrate complex. Another species is the product separate. So this enzyme substrate complex, there is a room for formation of enzyme substrate complex and also conversion of substrate into the product at the end. Right? Enzyme substrate complex formation and clearance of the substrate into the product. Both of these things continue in equal rate, and if both of this process continues in equal rate, there's a formation of enzyme substrate complex and then conversion of the substrate into the product. If both of these things are going on in equal rate, what we call it a steady state of the enzyme reaction. And what we mean by steady state of the enzyme reaction? Steady state of enzyme reaction, steady state of enzyme reaction says this important idea that the formation of ES and the clearance of ES are equal. That's the idea. And if formation and clearance are equal, then what we can say? Because if you take each of this site as a chemical reaction, and for each chemical reaction we have this reaction, the rate constant for every single reactions that we're looking here, right? We have the rate constant for that. So let's assume that the rate constant here for the association K1, and in this case the second rate constant K2. We can write it like this, right? Because here I'm not going to talk much about all the different mathematical aspect of the catalysis reaction, but it's a very simple term that if we assume that the first association the constant, reaction constant is K1, so the reversal of that is K minus 1, okay, and this one at the end, enzyme substrate to the product, is K2. Now remember one important thing, let me draw it fresh so that you can understand. Enzyme plus substrate can associate or can dissociate into enzyme substrate complex, but can be only one single arrow to produce a K product plus enzyme. This is K1, this is K minus 1, and this is K2. Okay, so now what was our assumption? Our assumption was we're looking at a steady state. Steady state means the formation of the enzyme substrate complex and the removal of the substrate into the product. Both are going on in the same rate. Okay. Now imagine one important thing at this point. In the steady state, this chemical reaction can reach a particular velocity, right? The velocity is marked with V0. That's the velocity that they are actually following. Now the question is how much velocity they can maximum achieve. We term it with Vmax, right? So normally at any point of the time, the enzymatic reaction, the velocity that they can take is V0, the initial velocity. Okay. On the other hand, the maximum velocity that they can reach is Vmax. But in steady state, try to understand, in steady state, this V0 equals to Vmax, isn't it? Because in steady state, there is no like it's always continuing at its full rate. So how exactly we know and how exactly we calculate the velocity of the enzyme reaction, and what are the factors this enzyme reaction depends for calculating the velocity? The velocity depends on the enzyme concentration, right? If we have more enzyme, they can achieve more velocity, right? Because I told you what enzyme is doing, lowering the activation energy, causing the process to go forward, right? So if you have more and more enzyme concentration, that means faster the reaction should occur. So now what we know, this clearance from ES to product and enzyme, the rate constant is K2. The rate constant is K2 here, right? So what we can tell from here is that this K2 is the actual reaction rate constant of conversion of substrate into the product. Normally without the enzyme this portion takes more time and it's actually slow, but with enzyme it's changing the time, it's changing the duration. So at this point of time, as we know that the V0 equals Vmax, and it depends, this velocity depends on the enzyme concentration. So what we can say here, what we can say that K2, or this reaction, the rate constant of conversion of enzyme substrate into the product and enzyme separately, equals to a value known as the maximum number of substrate molecules that can be converted to the product, right? So this K2 equivalent with the maximum number of substrate molecule that can be converted into the product per second, we call them Kcat. Kcat. So at this particular point, K2 equals Kcat. K2 equals Kcat. So now imagine one thing. Now imagine how to calculate the maximum velocity. We know maximum velocity, or the velocity of the enzyme reaction, depends on two things. What are those things? One is the enzyme concentration. Another one, this. First is the total enzyme concentration, and second one is this, the amount of substrate can be converted into the product per second, which is also known as the Kcat value, right? So this Kcat value, in the other hand, is known as the turnover number. So now if we want the formula of turnover number, Kcat equals to, it will be Vmax divided by total enzyme concentration. That will be the calculation for turnover number. So Kcat, or turnover number, equals to Vmax by total enzyme concentration, right? What is its unit? Per second, per unit time. So in this case, per second. Okay. So Kcat, or turnover number, is the maximum number of substrate molecules converted to product per second by each active site of the enzyme. Got it? The maximum number of substrate molecules converted to product per second by each active site of an enzyme. So if number of active sites are increased, the Kcat value will also increase, right? If the enzyme concentration increases, the total enzyme concentration, in this case, it also depends on, because you know the total enzyme concentration is linked with the maximum velocity that they can achieve. Now another thing, if I ask you, if we know that the Kcat is Vmax by ET, on the other hand, what we can say, inverse of Kcat, that is 1 by Kcat, equals to what? The total time taken for the enzyme to convert one substrate molecule to one product. Got it? So if we each inverse the Kcat value, we'll get the total number of time, or amount of time required for converting one substrate into one product molecule. So as we now know the Kcat, there is another term, catalytic efficiency. What is catalytic efficiency? Kcat by Km. Now what is this Km? What is the Km? Km is the amount of substrate. It's the substrate concentration at which the enzyme reaction reaches half the maximum velocity. So what Km signifies? What Km signifies? The affinity. It signifies the affinity of enzyme towards substrate. Affinity of enzymes towards its substrate. So the more the Km value, the less chance that they can bind more eagerly. So now think about this term catalytic efficiency. The better the kcat, the more the kcat, the higher the catalytic efficiency. And the less the Km, the higher the catalytic efficiency. So less Km means more affinity, that will increase the product formation, and higher kcat, that means higher turnover number, converting more substrate into the product per second time by the active site of an enzyme. Does it make sense? It does. These are the idea of any chemical, any enzymatic reaction. Okay, this is the idea of any chemical reactions that we know. Unit So think about what can be the unit, because you know the catalytic the kcat unit. Which part you want me to explain? Last part means which part? Catalytic efficiency and affinity. The term affinity I form and I told this term later. First I told you like what is Km in terms of real example. The Km will be, if I show you a graph, let's assume, and you started with this chemical reaction. Let's say, let me draw this in any other enzymatic reaction, and in an enzymatic reaction we put the substrate concentration in the x-axis, and let's put the velocity in the y-axis. Okay, so what we can see here, it starts from some point of here and goes something like this. You see here, substrate concentration in x-axis and the velocity in y-axis. So substrate concentration, as we are changing the substrate concentration, the velocity is continue to increase. That is when we assume that the enzyme substrate complex is at steady state. That means the formation of enzyme substrate complex and clearance of the substrate into the product are almost equal, are the same. Okay, at this particular point. So if you continue to increase the substrate concentration, as the enzyme's active sites are free, they will continue to bind to the substrate and convert them into the product. So it will continue to rise the velocity until a specific point, because once they reach a specific point, even after that, even if we increase the substrate concentration, the velocity of the enzyme will not increase more, because at this point all the enzyme's active site are saturated with their corresponding substrate. Okay, now at this point, if we continue to go at this particular velocity, where you can see, if this is the maximum velocity, because that can be achieved, the maximum velocity that can be achieved at a specific substrate concentration, and which is reached some point here, this high substrate concentration. Now this is the half of this maximum velocity. So now if I drag this line to the substrate concentration, we find the substrate concentration value, we call it Km. So that's what we know. Km is the substrate concentration for which half of the maximum velocity can be achieved. Okay. In this graph, you won't get an idea about the catalytic efficiency. Catalytic efficiency is a term, or catalytic efficiency is a result of involving both turnover number and Km. Now imagine one important thing. If the Km is high, what does that mean? That means to achieve half of the maximum velocity, you need more substrate. And if Km is less, to achieve the same velocity, you need less substrate. So which one is better? Low Km in terms of a chemical reaction. Low Km means it takes less substrate concentration to reach the half of the maximum velocity. So the reaction is working faster. That's why low Km is always desired. That's why we tag Km with affinity. The lower the Km, the higher the affinity. The higher the Km, the lower the affinity. On the other hand, Kcat is a separate unit. That is a turnover number. And I told you, turnover number means if this is an enzyme and this is the active site, okay, and substrate is now binding to the active site, and the substrate will be converted to the product by the active site. So it's actually the total number of substrate converted to the product by these active sites per second. That is the turnover number. So actually this catalytic efficiency depends on this turnover number by Km or affinity, right? So actually more the affinity or more the Kcat, the better the enzyme for its reaction. So if an enzyme has more turnover number, high turnover number and a higher affinity, that particular enzyme is a better enzyme. Okay. That is simply the idea of Michaelis-Menten. And Michaelis-Menten provided us the formula, the equation, Michaelis-Menten equation. That equation also states this simple idea, that if we assume that the enzyme substrate complex in a steady state, then we can easily find the enzyme reaction, and then what we can say, we can also find out the catalytic efficiency of the reaction. We can also find out the growth and the maximum velocity that the enzyme can achieve for converting the substrate into the product. If you look at here, whatever part we talked till now, I told you like the most important job an enzyme has is bringing the substrates or reactants close enough to each other so that they can establish the chemical reaction properly. If you remember, in the module 3 we understood the process of nucleophilic attack, and in those cases I told you as well, like the structures brought close to each other, the active site attacks the other one, the hydroxyl group, the hydroxyl acts as a nucleophile, attacks the neighboring group, and then the chemical reactions occur. So actually the reactions occur always like the same process. It's a nucleophilic attack, electrophiles formation, or any other process, the formation of electrostatic interactions, charged the dipole interactions, and all these interactions are going on to bring all the other molecules close to each other, joining them together covalently or non-covalently. But what enzymes can do is bringing them close to each other so that they can establish the task. So it becomes a lot easier. So in any case, this picture is a lot like, it's a very common picture, you probably have seen thousands of times. It states that the energy of reactants and the energy of products, and the energy of products are always less, because the energy is released somehow at this, because it's exothermic in reaction, the energy is removed at the end. So if and only if the energy of the products are less than the energy of the reactants, then only that reaction can proceed in the forward direction. That's the law of thermodynamics, because the difference between the energy of reactants and the energy of the product is known as the del G. So if the del G value is negative, that is the energy of the product is less compared to the energy of the reactant, then only the reaction will go forward. Otherwise, the reaction may not go forward. That is the simple stuff that we know. But to go from this energy of reactants to the energy of the products, there is this activation energy or hurdle that is present in between. You know why this activation energy, and why it's so much without the enzyme? Because you can see the without the enzyme, this red curve, with the enzyme, this black curve. So why without the enzyme we have the high activation energy, and with the enzyme the activation energy drops significantly? The reason behind this, what is the reason why without the enzyme we have a high activation energy, and why we need to go for this high energy barrier? Because, you know, as I told you earlier again, that the reactants are not closely placed. The reactants are distantly apart, and there are different interactions going on in between them. So they need to invest a lot more energy to initiate the process of the chemical reaction, initiate any kind of modification to the what we can say molecule, to the reactants, to convert it into the product. Because, you know, if you look at the structure of the reactants and the structure of the product, you will see the difference, because structure of the product is completely different than the structures of reactants. So when an enzyme is present, brings them close to each other, they need less energy, they need to work less, because now they are in close proximity, they can form bonds, they can break bonds very fast and very accurately. That's why it reduces the activation energy there. So this, in a sense, we can say that at the very beginning we have the enzyme substrate separately, then we have enzyme substrate complex. This formation of enzyme substrate complex actually lowers down the activation energy of the reaction, and then we can at the end receive the end products, that is product and the enzyme separately. And we know that the enzymes that's formed at this particular point, this enzyme, although the enzyme is involved, in reality the enzyme is involved with the reactants. They can form bonds with reactants and change and modify themselves a little bit, but whatever modification is done at the end, the enzyme will remain as it is. It can interact with formation of any kind of bonds and breakage of any kind of bonds, but ultimately at the end of the reaction it should remain as it is, without any change, without further alteration. Now we will see this representation of enzyme reaction, the representation with the two separate ideas. The idea of, in this case, the Michaelis-Menten equation, and the other hand is the another type of method that we'll see, the Lineweaver-Burke plot. So two types of plot that we actually see, Michaelis-Menten plot and Lineweaver-Burke plot. And why so much difference in both the plots? Let's look at it in a moment. I already provide you this idea of this particular graph, substrate concentration in x-axis and velocity in the y-axis. And you know, as we know the steady state assumption, we know the more the substrate concentration, the faster the reaction is working, so the more the velocity that's achieving. So it's continue to achieve the maximum velocity slowly, so continue to grow, and the substrate and enzymes are combining with themselves and converted to the product is increasing over time, but it will reach the maximum velocity. Maximum velocity is a situation where all the enzymes are saturated with the substrates, no further enzyme active site is free, so it's not being converted. Now if you continue to supply new enzyme, then you will see this velocity is going to be increased far away. Now if you look at the half of the maximum velocity, you will see only very few less substrate concentration is required to reach the maximum velocity. Then a lot of other enzyme concentration is required to reach the full velocity. But at the very beginning, the number of substrate, or actually the concentration of substrate that helps the enzyme to reach the half Vmax is known as the Km value here. Okay, so this idea is represented here with this graph, and this is known as the Michaelis-Menten plot. Okay, it's a simple drawing, simple plot, and we will also see different kind of enzyme inhibition utilizing this plot itself. But you know, this plot is not a straight line. You know, there is a formation of this the structures, because ultimately at the end it hits the saturation curve and stuff like that. So it's very hard and difficult for us to explain the competitive and non-competitive inhibition with the help of this graph always. So that's why we altered this graph. We altered it into a different kind of graph known as a Lineweaver-Burk plot. In the Lineweaver-Burk plot, the value for each of these reactions, the values are taken in log. So as we take the logarithm of the value, it will always give us a straight line in the graph, but these lines are never continuing through this y-axis. It's actually intersecting the y-axis as well as the x-axis. Both the axes are actually, this graph is intersecting. So those regions either intersecting in the y-axis and x-axis are known for different purpose here. For example, if you look at this Lineweaver-Burk plot, the portion where it intersects y is known as 1 by velocity, or so inverse of the velocity, 1 by v max, 1 by v, whatever it is. When it's intersecting the x-axis, that is known as 1 by km value, and as it's going left hand side of the 0 or the start side, we call it the value in the minus, so minus 1 by km value in the left hand side, plus 1 by km in the right hand side. That's the idea here. So actually this graph is plotted based on the inverse of substrate concentration, inverse of the velocity. That's how we'll get the maximum velocity as well as the 1 by km value intersecting the x-axis, and maximum velocity 1 by maximum velocity intersecting the y-axis. That's the idea. So we will see now, utilizing this Lineweaver-Burk plot, it's a lot easy for us to understand all the three types of enzyme inhibitions, because while we start talking about the inhibitions, you will see why it is so much important. So let's look at here about the enzyme inhibition, and there are these three types of enzyme inhibitions that are involved. One type of inhibition is known as a competitive inhibition, the second type is non-competitive, and the third one is uncompetitive. These are the three types that we generally talk about here. In the competitive inhibition, and on all these three types of inhibition, the scenario is completely separate. In competitive inhibition, as the term suggests, competitive, that means the inhibitor itself is competing for the binding with the enzyme along with the substrate, because substrate and inhibitor both are competing with each other to bind to the active site of the enzyme. Because this inhibitor resembling the structure of the active site, so inhibitor binds to the active site properly. Once it binds to the active site properly, then what it does, it will not allow the substrate to bind. So either a substrate or inhibitor can only bind to the active site. Okay, so what we can see here, that between the race between the substrate and inhibitor, anyone can win. So it depends on the concentration of the substrate itself, isn't it? So if the substrate concentration is far greater than the enzyme inhibitor concentration, then the substrate will always bind. There will be less chance of inhibitor binding and the enzyme reaction will work properly. So no matter what situation it is, if we provide more and more substrate, the competitive inhibition can be revived, right? It can revive, we can reverse the competitive inhibition. It only depends on the substrate concentration. So if you get enough substrate concentration, it will be reversed. So now if you look at here in the Lineweaver-Burk plot, how it looks like, because Lineweaver plot, what we know is simply the y-intersect and x-intersect, and the y-intersect is v max, right? 1 by v max. So this value of 1 by v max. So the question is, in competitive inhibition, can we reach maximum velocity which we used to reach without an inhibitor? Can we reach it? Can we reach maximum velocity? The same maximum velocity that we used to reach without the inhibitor. Actually, yes, we can reach maximum velocity. Why? Because if we increase the substrate concentration, then we can reach the maximum velocity at very high substrate concentration, where let's say for example For every one inhibitor, there are 500 substrate. At this particular level, we can still reach the maximum velocity. That's why if you look at this picture of Lineweaver plot, you will see that the Vmax, all of the data, all of these plots are intersecting in same point. That means Vmax is unchanged. It's not changing. As you can see here, we are continuously increasing the inhibitor. So this is without the inhibitor, this is with inhibitor, and this is even higher inhibitor concentration. This is also with inhibitor but less. We are going higher of the inhibitor concentration, but still you can see the Vmax is unaltered. What is changing here is the Km. That is one thing that will alter. What will happen to the Km? What's happening to the Km here? Just 1 minute. Give me 1 minute. So whether Km is increasing or decreasing, what do you think? Because you know this is 1 by Km minus, and what we are seeing actually, the as we are increasing the inhibitor concentration, this graph is going towards 0. So what's happening? Increasing. So imagine one thing, we are going towards 0 and the value of Km given is like this. So what do you think? Still we have two separate options there. Why? Now if I show you this picture, it will be more clear. If you look at here, this one, what do you think here in this picture? This is a Michaelis-Menten plot. Without any inhibitor is the pink one, with competitive inhibition with the blue one. You can see here, with the pink one the Km value is here, with the blue one Km value is here. So what's happening? Increasing. Right? Because imagine one thing, now if you look at the second type, is this one is the non-competitive inhibitor. We are looking at this process in contrast in the next slide, okay, where you'll see what is happening to each of these parameters. But if you look at the three separate things like non-competitive inhibitor and uncompetitive inhibition as well. Non-competitive inhibition, it's when the enzyme is free, but you know in this case the enzyme does not have only one active site, it has more than one active site. More than one active site. So in more than one active site, that's also known as allosteric site, right? So substrate can bind to the substrate binding site, and inhibitor can bind to the inhibitor binding site or allosteric site. So now the question is, in this case the enzyme can bind to either substrate, or the enzyme can bind to either inhibitor, or the enzyme can bind to both substrate and inhibitor at the same time. So if inhibitor is attached to any of their allosteric sites, it will not allow the enzyme to convert the substrate into the product. Now the question is whether we can achieve maximum velocity. Can we achieve it? No. We cannot achieve maximum velocity. So what will happen to maximum velocity? It will decrease. So the maximum velocity, whatever it was earlier, now it will be less. So if you look at the Lineweaver plot again, you can see the maximum velocity, the value for the velocity 1 by Vmax is changing. So at this particular point, increasing, the maximum velocity is also decreasing. Now what is happening to the Km? It's fixed. There is no change to Km. It's constant. Now the third one is uncompetitive inhibition. So what is uncompetitive inhibition? In this case, the inhibitor can only bind with the enzyme substrate complex. It does not bind to free enzyme, it does not bind to free substrate. It can only bind to the enzyme substrate complex. Right? Now if you look at all these three things, because now if you imagine, if you look at this graph, Lineweaver plot, what will happen to this plot in case of uncompetitive inhibition? You can see here that the value for 1 by minus 1 by Km is shifting to the left. While what's happening to the Vmax? Also decreasing. Because again, what will happen to the Vmax? Because we cannot reach Vmax. Now the fundamental difference between uncompetitive and non-competitive inhibition is, in non-competitive inhibition, the inhibitor can bind to either enzyme, free enzyme, or it can bind to the enzyme substrate complex. But in case of uncompetitive inhibitor only binds to the enzyme substrate complex. So now if we look at here, in case of competitive inhibition, what's happening to the Vmax? At sufficient substrate, Vmax can be reached. So there is no change in Vmax. In non-competitive, decreases. In uncompetitive, decreases. What's happening to the Km? For competitive inhibition, increasing. For non-competitive inhibition, unaffected. For uncompetitive, decreasing. Now the question is, in all these three types of inhibition, if we flood the reactants with substrate, can we reverse the inhibitor effect? For competitive inhibition, yes, we can reverse it, because with very high substrate concentration, we can reverse the process. In non-competitive inhibition, we cannot reverse the process of inhibition. In uncompetitive, we cannot reverse the process of inhibition. And the fourth kind is known as a mixed type, which is not shown here, which is not a unique type of inhibition process though. But you remember, in mixed inhibition, the binding site of inhibitor is separate site from the active site to either a free enzyme or enzyme substrate complex, which is similar with that of the non-competitive inhibition we saw. What's happening to the Vmax? It will always decrease, and that what happens to the KM, it also either decrease or increase. Okay. But if you look at these three pictures, that's why I told you, with the help of Lineweaver-Burke plot, we can actually visualize the changes between the enzyme inhibition of competitive, uncompetitive and non-competitive type. That's why we utilize this Lineweaver-Burke plot for analyzing the enzyme inhibition. We can also check this thing with the help of the Michaelis-Menten plot. Earlier we saw here like this for competitive and uncompetitive, as you can see. But still, it's better understood with the Lineweaver-Burke plot. Mixed inhibition has more similarity with that of that because mixed inhibition means enzyme can bind to the free inhibitor, converting enzyme inhibitor complex, or enzyme substrate can bind to the inhibitor, form enzyme substrate inhibitor complex. So whenever they form this enzyme substrate inhibitor complex, it's fixed. It can no longer convert the substrate into the product. And the difference between the competitive inhibition and mixed inhibition in terms of Km and Vmax. In competitive inhibition, Vmax can be achieved, it's unaltered. Km increases. In case of mixed inhibition also Km increases, but the Vmax goes down. That's the only difference. Because in that case, an inhibitor can bind to the free enzyme. So once inhibitor binds to the free enzyme, inhibitor binding site, it alters the structure of the enzyme, and that ultimately changes the turnover number of that enzyme. And as the turnover number is altered, catalytic efficiency also altered for that enzyme. Now let's look at the idea of isozyme and allozyme. As we're talking about enzymes, isozymes are genetically controlled variants of enzymes which have various metabolic functions. When they express themselves in different regions of the cell. It's not only common in plants, it's also common in animals and plants both. And there are these two terms, isozyme and allozyme. And as you say that iso means similar, allo means allelic variants. So isozymes means homologous proteins coded by different loci, while allozyme are the allelic variants of a particular protein formed at the same loci. So if you look at here in this particular picture, you see the two set of the gene A and gene B, the set of it. So set for gene A and set for gene B, two separate genes are there. So two separate loci are there, E1 and E2. E1 is a different loci, E2 is a different loci because they belong to the two separate set of the chromosome. So E1 and E2 both producing enzymes like enzyme 1 and enzyme 2, they will be known as their isozyme because they are formed from the homologous pair, because both of them, gene A, gene B, that's a pair, from one from father, one from the mother. So they are producing themselves from the homologous pair. That is why we call them isozyme. Although they are coded from different loci, but they are produced from the same homologous proteins, right? Are coded by the same homologous gene. What are allozymes? Now imagine the thing. Allozyme means in the same loci. If you look at the same loci here, in the same gene, let's say gene A, there are two alleles that are different. So two alleles are different, so the proteins will be produced from there, all very very much similar, but as they are produced from two separate allelic variants, we call them allozymes. Okay, so simply allozymes means proteins produced from here and here. These are allozymes. Produced from here and here will be isozymes. Few such examples. For example, in humans we can check lactose dehydrogenase. Lactate dehydrogenase, there are different variants, LDH 1, 2, 3, 4, 5, 6 different variants, and you can see here that they have different allelic variation. Like these are actually isozymes, so they are produced from different homologous regions, from different loci. So let's say LDH 1 occurs in myocardium, LDH 4 occurs in muscle and liver, LDH 3 is actually produced in acute leukemia, and things like that. Okay. Now, as I told you, there will be few things that you need to remember, like from all these macromolecules portions, like carbohydrates, proteins, especially carbohydrates and proteins, a little less in the part of the nucleic acids though. But you need to remember their like structures and few things regarding their stereochemistry. And one of these things are these disaccharides. You need to know the example of the disaccharides out there, okay, and the bonds that they form between them, what kind of bonds are there, and what are the properties of the disaccharides. For example, sucrose is produced from glucose and fructose, lactose is from galactose and glucose, maltose is from glucose and glucose. But these bonds are different. Sometimes a bond is alpha 1,4 or beta 1,4, beta 1,2. These are the different types of link that we see. For example, if you look at lactose, it's between the bond between galactose and glucose, and it's a beta 1,4 kind of bonding here. Beta D galactose with the alpha D glucose. While in maltose, it's a alpha 1,4 for both of it. And for glucose fructose binding for sucrose, this is 1,2 bonding, alpha glucose and beta fructose. So this is the type of different bonds that we know. And another term that you also need to know is a reducing sugar. What is known as a reducing sugar? Any sugar that is capable of acting as a reducing agent, because it has either a free aldehyde group or a free ketone group. That sugar is known as a reducing sugar. Now, it's fine for calculating a reducing sugar which is like monosaccharide, which is only a single unit of the sugar. But if it's disaccharide, that's really sometimes you need to remember that which of the disaccharides are reducing and non-reducing. For example, in the disaccharides, lactose, maltose, these are reducing type, but sucrose is a non-reducing type, because sucrose does not have in this case any kind of free aldehyde or ketone group due to the 1,2 linkage. On the other hand, whenever the link is 1,4 link, there is a chance of this formation of and actually the chance of having the free hydroxyl group, free aldehyde or ketone group. Now there are a few terms that I want you to remember from where a question can be there, like isomers, apimers, anomers. These are the terms you need to know about them in reference to the structure of monosaccharides. So what is isomer? Isomers are the compound, they have same chemical formula. For example, glucose, galactose, fructose, mannose, all of them have same chemical formula, and that is C6H12O6. That is isomers. All of them same chemical formula. What are apimers? Two isomers that they differ in configuration around only one specific carbon atom other than the carbonyl carbon are known as apimers, because carbonyl carbon is separate. Without the carbonyl carbon, in all this case, which is carbonyl carbon? This is CH or CH group, whatever it is. This is the carbonyl carbon. Apart from the carbonyl group, wherever the other carbons are present, among the other carbons, the change in configuration around any single carbon apart from the carbonyl carbon will term the other saccharides as their apimers. For example, D-mannose, D-glucose, D-galactose, whatever things are there. If you compare between the glucose and mannose, what we can see, glucose and mannose in the second carbon. This is the carbon. If you look at the second carbon here, apart from the carbonyl carbon, this is the second carbon. In case of mannose, it's H in the left side, OH in the right side. But in case of glucose, OH in the left side, H in the right side. It's not like the whole structure is a mirror or a mirror image. It's not like that. But they have altered in one particular carbon. So that's why we call them epimers at the C2. The same thing, the epimers at C4, glucose and galactose. The epimers at C4, because the carbon number 4 are different. As you can see, H left, OH right for glucose, OH left, H right for galactose. Those will be known as the epimers. So epimers of carbon 4 is glucose with galactose, and carbon 2 is mannose with glucose. Can't you see the screen? If there is a problem, you should rejoin. Now, what is enantiomers? Enantiomers, a pair of structures that are mirror images of each other in regard to the asymmetric carbon atom present in their molecules. If you look at the structure here, all the asymmetric carbon atom, that means this one, this one, this one, this one, structures utilizing this carbon atom, and if you compare them, they will be appearing like a mirror image of one another. That is known as enantiomers. See, hydroxyl in the left, right, so in this case left hydrogen, right hydrogen, they will be known as the enantiomers. So in all this case, we term them either D or L depending upon the position of the OH group on the last asymmetric carbon. So in this case, you see on the last asymmetric carbon is this one, and the hydroxyl is placed on the right side, so we call them D glucose. And on the other picture, this one, the hydroxyl is placed in the left side, L glucose. Another term, anomers. So we talked about isomer, epimer, enantiomers, now anomers. Anomers mean those two stereoisomers, of any given sugar that differ only in the configuration about their carbonyl carbon. That means between two sugar, they have same chemical formula, differs only in one configuration at the carbonyl carbon atom only. And based on that, we call them alpha or beta. So in alpha isomer has the hydrogen atom. If you look at here, the alpha isomer, the hydrogen atom in the C1, so this is the carbon number 1, the hydrogen atom is out of the plane, on the top, above. In beta, the hydrogen atom in the C1 is below the ground, below the plane. So if you consider a plane, the hydrogen atom can be above the plane or below the plane. That's anomers. So what's the difference between the anomers and epimers? Anomers and epimers? In epimers, it can be difference in configuration in any of the carbon apart from the carbonyl carbon. While in anomers, it's only the change around the carbonyl carbon. And it can be visible or it can be explained well if we look at the three-dimensional structures of either the hydrogen is placed above the plane or below the plane. Now let's talk about a little bit about the protein structure morphology. In protein structure morphology, we know the protein structural hierarchy and starts from the primary structure, then secondary structure, tertiary structure, quaternary structure. Primary structure is the one where all the amino acids are linked with one another with the help of peptide bond. And we know the peptide bond formed during the elongation phase of protein synthesis, and this process is a transpeptidase reaction. Two nearby amino acids are linked with one another. So once all the amino acids are linked with one another, we form a long chain of amino acids. That is the primary structure of amino acid. Then that primary structure of amino acid, that structure, those amino acids that are linked with one another form this structure, and those amino acids will be now linked with one another, forming bonds with one another to form secondary structure. So what is this secondary structure? What is the secondary structure? The secondary structure is the characteristic structures that occur in many proteins, that is alpha helix, beta sheets. These are the two major secondary structures that we see. And what is the difference between the primary structure and secondary structure? Because the primary structure is only the amino acids linked with one another with the peptide bond. But when those amino acid structures, they are placed close to each other and they form the bonds with each other, the hydrogen bonds with each other with the help of the carbonyl carbon, the oxygen from there, the carbonyl group, and the hydrogen from the NH group from the neighboring amino acid. They interact with themselves to form hydrogen bonds to make the secondary structures. So secondary structures are the unit structure that will be required to make the tertiary structure of the protein. And tertiary structure is the one where we have the three-dimensional folding of a protein. Three-dimensional folding of a protein ultimately dictates the function of the protein, because once the protein is properly folded, then only that folded structure of the protein can act as a functional unit of an enzyme. Now this tertiary structure is the minimum structure required for any protein to work and function. Now one particular protein may have multiple tertiary structure linked to one another. Then in that case it will be known as a quaternary structure. If we look at here the protein structure hierarchy, this is the primary amino acid chains linked with the peptide bonds. Secondary structures, alpha helix, beta sheet, and the tertiary structure, then quaternary structure, multiple tertiary structure linked with one another. Now in all this case, these are the two most important, the secondary structure, alpha helix and sheet. This alpha helix or beta sheet, that they will fold, they will interact with themselves to form complex three-dimensional structures of the protein. Because if you look at the structures between beta sheet and alpha helix, one is helical, another one is a sheet-like structure. The difference between the two is both of them, they form the hydrogen bond by the neighboring amino acid. The carbonyl group from one and the NH group from the neighbor. Carbonyl from one, NH from the neighbor. And that's how they interact with themselves. But the interaction pattern is different. In alpha helix, if you look at here, the alpha helix, the carbonyl carbon is linking to the NH there, the hydrogen of the NH. This is how the hydrogen bonding is formed. On the other hand, the beta sheet, it's also the similar reaction like carbonyl carbon and the oxygen from the carbonyl carbon with the hydrogen neighboring. They are binding with themselves. But the difference here is the number of amino acid that they linked with one another. In case of alpha helix, if you look at here, in case of alpha helix, it's known as simply that I and I plus 4. So I means the first amino acid, and after the fourth one, the fourth amino acid there will bind to the first amino acid with hydrogen bonding. So this pattern of I and I plus 4 continues. This is the typical structure of an alpha helix. That's why they form the helical structure, because first at the beginning the first amino acid known as the I, and then sequentially those amino acids are linked 2, 3, and the fourth one plus 4, the fourth amino acid, and then NH from the fourth amino acid and O here from the carbonyl group of the first amino acid interacts with the hydrogen bonding. That's how they continue to arrange the same way and form a helix. While in beta sheet that's not the case. In beta sheet the interactions are far distantly placed amino acids, because in alpha helix the interactions are between very close, either I plus 4 or I plus 3. In sometimes you will see the helical structures formed without I plus 4, but I plus 3 after every third amino acids. After every 3 amino acids, so the first one and after the third, the fourth one interact with themselves, or after the first one and after the fourth one they interact with themselves. So either I plus 4 or I plus 3 helix. So if it's I plus 3, we call them 310 helix, while if it's I plus 4, we call them the normal alpha helix. Okay. I means nothing here. I means the start, the initial. You can also say X, Z, Y, whatever it is. It is the initial amino acid to begin with, and then continue with the others. Plus 3 means after the third, plus 4 after the fourth, like that. So it's very closely linked. 1 with 1 plus 3, 1 with 2. 1 plus 3, no, it's not exceptional case, but it's rare. It's not that common. I plus 4 is the most common type, that is known as the basic alpha helix. Whatever we talk about alpha helix, we mean I plus 4. But in some time, we also have the same helical structures, but with I plus 3 instead of I plus 4. Okay. And in that case, you can see here that this beta sheet, on the other hand, if you look, the beta sheet, in case of beta sheets, multiple amino acids, they are linked with each other, but they are distantly related. That is one particular, if we start numbering the amino acids from here, the amino acid number I, then I plus, this is the first one, second, third, fourth, 5, 6. So you can see the third one is interacting with somewhat 26 number amino acid neighboring. So the interactions are placed far distant apart, from the third one with 26, like fifth one with 50th. That's all. That's the difference of arrangement between both the structures. But what is very important here is this alpha helix and beta sheet structures actually dictate what kind of protein is going to form, because I already told you while discussing the membrane proteins, right? When we discussed about the membrane proteins, I told you like this alpha helix and beta sheet, both of these structures, they are like sometimes in few types of protein structures the alpha helix is more familiar, or in some other type of proteins the beta sheet is more familiar. So generally, you know, alpha helical proteins are most common in case of membrane proteins and structures, although beta sheets are also found. The same idea is prescribed by a specific angle known as the dihedral angle, which helps us to identify and understand the interactions between the amino acids nearby, and due to their R group in the back, what we can say, apart from the backbone of the amino acid, the R group, changing of the R group, because you know R group can be as simple as an hydrogen, it can be very complex like a proline has. So there are different types of R groups, and depending upon those different R groups, there's a maximum configuration allowance that can be provided to any of these two neighboring amino acids linked with one another. If you look at here, the two neighboring amino acids linked with one another, what we can see is NC alpha C. So this is the alpha carbon. Alpha carbon means this is the one with 4 separate bonds interacting with 4 separate other atoms there, and that is nitrogen here, nitrogen R group, the other hydrogen, and the C carbon. So NC alpha C, then again NC alpha C. This process continues in the peptide backbone. So what we know here is this NC alpha C, in this particular structure here of NC alpha C N, you can check one important thing, that the interaction between N and C alpha, and on the other hand the C alpha and C, from both of them they have an axis. This is the axis for NC alpha, this is the axis for C alpha C. Now rotation around the axis is possible. Rotation around both of these axes are possible if the groups attached in both this case, you know, CO group, the carbonyl group is present, and in this case the NH is present. but this R group is the bulkier one, or it can be a very similar one. If it's bulkier group, then this rotation is not much it will be prohibited, the rotation is not much allowed. But if it's a less complex group, then yes, the rotation is possible. But for both this axis, the rotation is possible. So for the rotation between n c alpha here, we call them this phi angle, and the c alpha c, we call them psi angle. So what are the different degrees of this angle? You know, the rotation is possible 360 degree, right, ranging from the point. And if we maintain one particular point, we can say in one side, let's say on the top of this, because you know the structure is three-dimensional, so imagine a plane. So rotation around the top of the plane, let's assume 0 degree to plus 180 degree, and on the other hand 0 degree to minus 180 degree, ultimately giving us total 360 degree rotation around the axis for both phi angle and psi angle. Try to imagine that the structure is rotating, but the question is this rotation around this axis, this rotation around this structure depends solely on the structure of the amino acids and what kind of amino acids are present and how they are assembled. Because you know, in alpha helix we need to assemble the amino acids in very close proximity with 1 plus 4 or 1 plus 3, but in beta sheet we can assemble them in a long spreadsheet like structures. That's the difference, that's we can figure out. So Ramachandran and his colleague, they discovered a way that they have understood this process, and what they tried to figure out is the value for both phi and psi angle for which the maximum rotation allowance is provided for any particular structure, either alpha helix or beta sheet. Okay, because you know, for a specific value of phi and psi, the structure is allowed only for alpha helix to rotate or only for beta helix to form. So this idea is captured with a plot known as Ramachandran plot. So in this Ramachandran plot, what is provided? They provided us information. They provided us the information of all this value of phi and psi angle for which an alpha helix structure is favored or a beta sheet structure is favored, whether a right-handed helix is favored or a left-handed helix is favored, whether a beta turn is favored. So these are the type of things, predictions that we can make. So once we know the phi and psi angle of a protein, we can also predict whether those amino acids are going to form more alpha helix or they are going to form more beta sheet, or if they are going to form the alpha helix, whether the helix will be left-handed or right-handed. These are the things that we can understand. These are the things that we can discuss with the Ramachandran plot. That's why this Ramachandran plot is really, really important. So now if we zoom into the Ramachandran plot here in this next picture, you can see here this Ramachandran plot is a way to visualize all the energetically favored or energetically allowed regions for the backbone of all these angles that we talked about, that the phi and psi, which are also known as the dihedral angles. Okay, for all this backbone of the dihedral angles for phi and psi values are allowed to have either a beta sheet structure or alpha helix structure. Okay, so if you look at here in this plot, that it's actually quadrant plot. This whole graph is divided. This whole plot is divided into 4 separate quadrants. In the x-axis we all have the 0. And considering minus 180 in the left hand side, plus 180 in the right hand side. And again in the y-axis it starts from 0, plus 180 in the top, minus 180 in the bottom. And what we are looking at here is that this beta sheet, so you can see most of the time if the value for the phi angle is in minus, especially minus 160, minus 170, while the psi angle is plus 160, plus 170, plus 180, exactly the opposite. In those conditions, those amino acid structures are favored to form beta sheets. Okay. Now on the other hand, if the value for psi is minus, but the value for phi is also minus, that they are going to form right-handed alpha helix. On the other hand, if the value for both are positive, if you look at here, both are positive. Value for phi and psi, both are positive. They used to form left-handed alpha helix. So in this case, just you need to remember left-handed alpha helix, right-handed alpha helix, beta sheets, where they actually present, because they can give you questions like they can ask you question from giving you the total phi and psi angles, and they will ask you to predict the type of structure that amino acid primary sequence can form. Okay. It's an assumption, because every single time they conducted this experiment of different structures of amino acids linked with one another, all the amino acid structures that are linked with one another, and then continue to check whether they are forming the alpha helix or whether they are forming the beta sheet. And they found out that whenever they provided the value of let's say both of them are in minus value, they are going to form the right-handed alpha helix. That's an observation that they can check. It's not like they're going to see this, or you can say it for sure, but yes, you can say that multiple times when they conducted experiment with the phi and psi both angles to be and the negative value, they are going to end up in forming the alpha the right-handed alpha helix structures. So actually it's nothing but representation of providing you the idea about the allowed regions for forming alpha helix or beta sheet by knowing the phi and psi angles, the dihedral angles between the protein backbone or amino acid backbone of the primary structure. Parallel and anti-parallel values are very hard to calculate, and it's although this picture is showing that parallel and anti-parallel almost superimposing with themselves, the difference between these two is also very much under the regulation, because when we are looking at these structures, the protein structures, the complex structure of the proteins, and actually in this case we are not only looking at the primary structures, we are also opting the structures, secondary structures and formation of secondary structures, then looking at the dihedral angles of the different amino acids placed in that secondary structures. So by looking at that structure, we can find a very little difference between the parallel and anti-parallel beta sheet in few parts only. Okay, because most of it, both of the cases, whether it's a parallel or anti-parallel beta sheet, it was found like the phi angle and the psi angle is going to be plus and the phi angle is going to be negative in value. And actually this will become more clear with this data that I'm showing you, because it's not about formation of alpha helix and beta sheet only knowing the values of phi and psi angle. It's also very important to know the relative frequency of amino acids and different structures. The relative frequencies of amino acid means because there are these 20 separate amino acids, we know, and among these 20 separate amino acids, not all the amino acids intend to form alpha helix, not all the amino acids intend to form beta sheet. There are few amino acids more found in alpha helix because structurally they are more allowed to form alpha helix, and there are few amino acids which are structurally more favored to form a beta sheet. Okay, so if you look at here, like glutamic acid, alanine, leucine, methionine, like few of them, especially glutamic acid and alanine, they are much dominant in alpha helix. You can see the relative abundance in alpha helix is more than 1, but in beta sheet is less than 1. On the other hand, if you go back here at this point, we can see that threonine, the abundance is more in beta sheet, less in alpha helix. Okay, tyrosine, cysteine, more in beta sheet, less in alpha helix. So there are this, we can say that some amino acids favor in forming structure alpha helix, some favor the structure in formation of beta sheet. You don't need to remember all the values, but just to get an idea about with changing amino acids, we can form different types of secondary structures. So I can tell you, like, if a protein is made more with tyrosine, cysteine, threonine, what we can say? We can predict that the protein is mostly to form a beta sheet, because for most of the times those amino acids are much allowed to form a beta sheet rather than an alpha helix. It's not likely that they are not going to form any alpha helix. They may form alpha helix, but they're much more favored to form beta sheet. Now let's talk about the protein domain and motif. What are these protein domain and motif? If we look at the units of a secondary structure is the actual what I can say, the organization that begins after the primary structure, because primary structure is nothing, it's so much arbitrary. There is nothing to be done with a primary structure in terms of a function of a protein. To initiate forming a functional unit, which is tertiary structure, by the way, the amino acid sequence need to form this secondary structure, and there are these two forms, alpha helix, beta sheet. But the arrangement of this alpha helix or beta sheet with one another, the interactions, there can be difference, and this different structure that is formed while repetitive secondary structures are linked with one another are known as super secondary structures. For example, 2 alpha helix, beta alpha beta, 2 beta, all of these structures are super secondary structures. That means multiple secondary structures are present at a time, side by side. That is super secondary structure. Okay. Now once they form this super secondary structure, then what we call them? The super secondary structures start to form different types of domains. What we mean by domain? Because there are these three terms: super secondary structure, domain, and motif. It's always confusing. So let's clear this doubt. Super secondary structure is while we place all the secondary structures linked with one another. That is the super secondary structure. 2 consecutive alpha, 2 consecutive beta, 1 alpha, 1 beta. And while we are forming the structures, it's not only how many alpha and how many beta are present which is important. It's also important, like, to know what is the sequence, what is the order. If it's alpha beta alpha, it's one type. If it's beta alpha beta, it's a separate type. So now if we're looking at multiple alpha linked with one another, alpha 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 alpha, that is one type. Beta alpha beta alpha beta, that is the second type, right? Beta 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 beta, it's a separate type. So repeatedly beta units continually. So these are known as the domain of the protein. The super secondary structures continue the repetition, that's the domain. But what is the motif? Because the domain and motif are used synonymously in many cases, but there is a very tiny difference between domain and motif. The domain is when we have multiple super secondary structures linked with one another. But what is motif? Motif is when we look at the pattern of the fold. That will be known as the motif. That will be known as a motif, So domain we can call like alpha 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 domain, or beta alpha beta alpha beta. It's also a domain. Beta 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 beta. It's another domain. But motif, we are calling motif what? Greek key is a motif. Rosman fold is a motif. As you can see, beta alpha beta, that's a Rosman fold. It's a motif. 4 betas together is a Greek key motif. And we are also calling them domain of a super secondary structure. So why two separate idea? When you say beta 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 beta, we call it the domain. It's a consecutive beta domain. Or you can say beta alpha, that's a consecutive domain. But when we call it motif, when we are looking at the folding pattern of this, an interactive pattern. For example, this 4 beta can form different structures. It can form a structure like this, right? First beta, second beta, third beta, fourth beta. But when it folds like this, first, second, third, fourth. Then this one, this is Greek key motif. So whenever we are adding order to it, whenever we are adding the sequence to it, how exactly it's folding, then we are talking about motif. Domain is only the structural part, the unit that we know. But motif is how that structure is organized. That is motif. Okay? So there are these two separate types of protein that can form about their structure. It can be either a fibrous or a globular. A fibrous structure means the long, thin structures. A globular structure is spherical, three-dimensional, large structures. And most of these globular structures of the protein, they are soluble proteins. For example, this hemoglobin is a globular protein. So in this globular protein, they need to interact to the hydrophilic solution all the time. So their surroundings are made, they should be made with hydrophilic amino acids. And the center is known as the hydrophobic core, and they should be prepared with hydrophobic amino acids. But the fibrous type of proteins, if you look at the fibrous type of proteins, they form this long thin chains, and those chains, especially now alpha helix, in most of the times they interact with themselves, either alpha helix or 310 helix. They interact with themselves with hydrogen bonding and many other type of interactions to hold that structure together to form a thin but very strong rope like structure. That is known as a fibrous protein. Example of fibrous protein: keratin, collagen. Example of globular protein: hemoglobin, insulin, and most of the other enzymes are acting as this globular protein. So this globular protein is soluble because they have the hydrophilic amino acid surroundings. Fibrous protein are insoluble, okay, because the job of fibrous proteins are different to provide structural strains. For example, collagen in bone and cartilage, keratin in fingernails and hair. On the other hand, this globular proteins, they always work in inside the cell, or let's say in case of the hemoglobin, they're working in the blood. And how the collagen is formed, we all know as an example of collagen formation. It's very similar with that of the intermediate filament. I already told you earlier. It formed like it produces the such multiple amino acids, primary structures are linked with one another. They interact with one another. We call them the nucleation. So once the nucleation is done with at least 3 such chains interacting with themselves, we call them the assembly of the chains. And then once multiple chains interact with each other, interwound with each other, we call them formation of procollagen, which is a predominant the preliminary form of the collagen. And then this procollagen is converted to collagen with a simple chemical modification. If we look at the collagen structure, you will find out the most of this collagen structure. This collagen structure is mainly formed with 3 amino acids, and the sequence is repetitively the similar: glycine, X and Y. What is X and Y? X denotes proline, Y is proline or hydroxyproline. So glycine, proline or hydroxyproline. That's the repeated sequence continue to be present. So approximately 33% glycine, 30% proline or hydroxyproline, and 37% any other peptides. That continues. 33% glycine, 30% proline or hydroxyproline, and 37% other peptides. That continues there. But most of the part they are made up with 30% and 37%. So 67% constituent of this glycine, proline and hydroxyproline. And it's always this particular structure, it's always are going to form more of a alpha helical structure, because with glycine, proline and hydroxyproline, the only way it can be possible to make a very thin alpha helical structure, because the glycine has a very small R group, that is H, and the proline has a very bulkier group, so it can prevent the steric hindrance, so as a result of which it can form an alpha helical structure. Without that, any other bulkier R group is not possible in the middle to form. this collagen structure. That's why it's always like glycine and proline that are found in this structure. Another thing that you should remember, I also told you in the earlier classes, that you need to remember the type of the amino acids which are hydrophilic and which are hydrophobic. So which go inside the protein, which stays outside. For example, valine, leucine, isoleucine, methionine, phenylalanine, they are mostly present in the protein interior, far away from the solvent molecules. While arginine, histidine, lysine, asparagine, aspartic acid and glutamic acid are typically located on the surroundings of the protein. Because all these things, lysine, histidine, arginine, aspartic acid, glutamic acid, they are also charged amino acids, and they are always interacting to the hydrophilic solution, where valine, leucine, isoleucine always favors the hydrophobic region of the protein. So you can look at the structural folding based on the hydrophobic nature of the proteins, and you go with the highly hydrophobic, moderately hydrophobic, mildly hydrophilic, and highly hydrophilic. The highly hydrophilic one will be the all charged one: arginine, lysine, aspartic acid, glutamic acid, glutamine, asparagine, histidine, all of them, they will be hydrophilic. Then moderately, the hydrophobic one will be tryptophan, cysteine, glycine, all these things. Highly hydrophobic will be isoleucine, valine, leucine, phenylalanine, methionine, alanine, all these examples. So you need to remember these parts, because this part and single letter code of all the amino acids are really important, because they can give you any kind of analytical question from this part. In this example, you can also see that the name of the group is given along with the characteristics and the example of amino acids belonging to the group, whether it's a basic, lysine, arginine, histidine, acidic, aspartic acid, glutamic acid, polar, the rest of the others, glycine, serine, threonine, cysteine, non-polar, alanine, valine, leucine, isoleucine, example. So this utilize or remember any of these tables. It will be beneficial for you to calculate if they ask you any question, because we already knew about the hydropathy plot, and we know with the help of hydropathy plot, which is also known as hydrophilicity plot, we can actually predict whether the primary sequence of amino acids, we can predict whether that sequence is going to form a membrane, an embedded membrane protein, or it's going to form a globular protein. It's really interesting. And we know that if it contains a large hydrophobic amino acid range, it's going to form only one single transmembrane protein, and if it has multiple sections of the hydrophobic range, it can form a multi-pass membrane protein at the same time. Now we'll talk about little bit about the globular protein, one example of the globular protein, which is hemoglobin, and a little bit about the myoglobin as well. And especially in this hemoglobin, you need to know the hemoglobin oxygen saturation curve. From the oxygen saturation curve, they ask questions every like it's very favorite area to ask question in CSIR net. So what's the job of hemoglobin? The role of hemoglobin is to bind oxygen and to carry oxygen in the blood and to remove the oxygen in the tissue, right? So all these things need to be done. Remember, so hemoglobin's job is to bind to the oxygen in lungs, carry the oxygen through blood, and release the oxygen in tissue. These are the three jobs. This is the complete work of an hemoglobin. So if hemoglobin fails to release oxygen to the tissue, then that's a problem. If it fails to bind to the oxygen in the lungs, that's also another big problem. So in every single time that we're looking, all this process need to be done properly. And that's why hemoglobin has this typical structure of a porphyrin ring. At the center, iron is present, and 4 such subunits are required to form a quadrant structure. So we call it the quaternary structure of a hemoglobin, because each of these subunits are a tertiary structure. So they're capable of doing their own function, but to increase the capability of oxygen binding affinity, hemoglobin utilizes 4 subunits linked with one another, and also these 4 subunits linked with one another carry more oxygen, bind with more oxygen, and the oxygen binding affinity is increased once one of the subunits is attached with the oxygen. That is known as a cooperative binding. That means among these 4 such subunits, if I show you here, these are these 4 subunits. So the first subunit binds with oxygen. That will increase the affinity of the second subunit to bind the oxygen. That will even further increase the third subunit to bind the oxygen, and that will even increase the force affinity to bind the oxygen. So slowly the affinity towards oxygen increased once oxygen molecule binds to one of the units, and that continues. So once the oxygen binding is properly done, then this hemoglobin can carry the oxygen through the blood as they present inside the red blood cell. And another very interesting fact is the structure of hemoglobin with chlorophyll. Both of them have a porphyrin ring, and both of them have this iron. Like, the only difference is the hemoglobin has iron in the center, in the core, while the chlorophyll have magnesium in the core. That's the only difference. But both of them plays a vital role in a life support forms, and both of them are the color of their life. Plant, it's green, provided by the chlorophyll, and in our case it's red, the color of our blood, provided by the red blood cell, provided by the hemoglobin itself. Now the question is, what is this oxygen dissociation curve, and why we are bothered about knowing the oxygen dissociation curve? As I told you, the job of the hemoglobin is to bind to the oxygen in lungs, carrying it through the blood, and removal of the oxygen to the tissue. This is the total cycle of hemoglobin, by taking oxygen from the lungs, removing the oxygen, and releasing the oxygen to the tissue. And the hemoglobin also binds with the carbon dioxide which was released by the cells, and then bring it to the lungs and remove it in the lungs through the expiration. So when you look at the oxygen dissociation curve, it gives us an idea about the affinity of hemoglobin to bind with an oxygen molecule. So the affinity of hemoglobin alters at different point of time at different parameters. So what are the parameters that alters the oxygen dissociation curve of hemoglobin? One such parameter says, the more the hemoglobin, the better the oxygen binding. Because if the more the hemoglobin, the more they can bind to the oxygen. This is one thing. And the more the oxygen pressure. So the partial pressure of oxygen plays a very vital role. If the partial pressure of oxygen is high, that means the hemoglobin will bind to the oxygen better way. Right? And if the partial pressure of oxygen is low, so the association will also be low. So the higher the partial pressure, the faster the association. So as you can see in this picture, we are going from this particular point through this x-axis, and we can see with increasing partial pressure of oxygen, the saturation curve is also going on top. And the y-axis, what we have, the percent saturation of hemoglobin. So the more the oxygen partial pressure, the more the hemoglobin is saturated. But after a particular point, the saturation curve, the percent saturation is also saturated. What does that mean? That means all the hemoglobin oxygen binding sites are gone. So literally there is no site free in the hemoglobin to bind with oxygen. So what we can see here, that after reaching a specific partial pressure of oxygen, when all the active site for binding with the oxygen is gone, all the oxygen binding sites are gone, then they reach a saturation. That is one thing. They'll reach a saturation there. Okay. So at this point you can check here, at this point of partial pressure of 11 kilopascal, all the hemoglobin active sites are saturated. All hemoglobin oxygen binding sites are saturated. So there is no further oxygen binding, even though we'll increase the partial pressure of oxygen. No benefit. Now the question is, why we are looking at this? Imagine one important thing, that this higher partial pressure is present in where? In the lungs, right? In lungs we have higher partial pressure. of oxygen because oxygen is constantly inserted into the lungs due to inspiration. So in lung we have higher partial pressure, so oxygen is continued to bind with hemoglobin, and that's supposed to happen. So in lung we need hemoglobin to bind with more and more oxygen, but in tissues we want the hemoglobin to release oxygen. So usually we know in tissues the oxygen partial pressure drops. That's one thing, because it's more of a carbon dioxide in tissue than the oxygen, because oxygen is being utilized in cellular respiration. They produce more and more carbon dioxide. So as they produce more and more carbon dioxide in cell and less partial pressure of oxygen, it causes the oxygen to dissociate from hemoglobin. And how the curve will look like? The curve will shift slightly to the right. Let's look at here. Imagine this idea and why this graph is shifted to the right. So this blue colored graph is showing the oxygen saturation curve while we are looking at lungs. But whenever we are looking the dissociation of oxygen, which is represented with this dotted red line, which is in cells, what we can see here is this decreasing in pH, increase in diphosphoglycerate, and increase in temperature. All these three things, they actually are in favor of dissociation of oxygen. So somehow if the pH is acidic, if they form lots of diphosphoglycerate or the bisphosphoglycerate, similar things, or at the higher temperatures, it will cause the hemoglobin to release the oxygen, and that is possible in tissue. So what happens in tissue? In tissue there is more carbon dioxide and less pH. That's what happens in tissue, and this is known as the Bohr effect. So actually Bohr effect means sliding of the hemoglobin oxygen saturation curve to the right. If we look at here, let's check this. So this is the Bohr effect, sliding of the oxygen saturation curve to the right. What does that signify? Because in the cell, in the tissue, we have low pH, that means more acidic, higher temperature, higher partial pressure of carbon dioxide, higher concentration of 2,3 ppg. So what is Bohr effect? Bohr effect states that the hemoglobin's oxygen binding affinity is inversely related to both the acidity and to the concentration of carbon dioxide. So this hemoglobin's oxygen binding affinity is inversely related to both to the acidity and to the concentration of carbon dioxide. Okay, and you can see here, for all these reasons, low pH, high temperature, more carbon dioxide, more metabolic wastes, this curve shifts to the right. And that's true, because if you look at here, the oxygen saturation curve starts here, the fully saturated oxygen at this point. Slowly, as you can see, if you're looking at the tissues, the partial pressure of oxygen drops, so the oxygen starts to be dissociated from the hemoglobin. It continues to dissociate till a particular very low level of oxygen partial pressure. Now another important thing is there. Let's assume you are running, you're doing a very hardcore exercise, and that exercise is burning a lot of oxygen. You're taking a lot of oxygen for the process. So as you're taking a lot of oxygen, you'll run out of the oxygen very, very fast, because there will be limitation of the oxygen that hemoglobins can actually bind to. Right? So you can see here, this is the hemoglobin's oxygen saturation curves. Looks like an S-shaped curve. So while the partial pressure of oxygen is 20 torr, over 80% of oxygen is dissociated, because you're burning it, you're taking a lot of oxygen at the very beginning. So at a pressure of 20, partial pressure 20 torr, 80% of oxygens are gone. You don't have oxygens left. The hemoglobin can't supply the oxygen properly. You're gasping, and that's the point. Your body demands more oxygen. So for that reason, our body has a second oxygen reservoir, and that is known as myoglobin. So what about the myoglobin? If you look at the myoglobin's oxygen dissociation curve, and in contrast with the hemoglobin's oxygen dissociation curve, what's the difference? You can check that in myoglobin, while the partial pressure of oxygen drops to 20 torr for both myoglobin and hemoglobin, 80% of the hemoglobin already dissociates all the oxygen. But myoglobin was 90% saturated with oxygen at that very low oxygen pressure. What does that signify? Even if you drop the oxygen partial pressure to 10 torr, 75% saturation still left in myoglobin. So what does that signify? It signifies that myoglobin can remain attached with oxygen for a long period of time compared to the hemoglobin. Hemoglobin's job is to release oxygen faster, to pick and bind with oxygen faster and release it fast when they get acidic environment or higher carbon dioxide partial pressure or higher DPG or high temperature. But myoglobin's job is to remain attached to the oxygen. So when the hemoglobin run out of oxygen supply, myoglobin can continue to provide oxygen to the muscle, and the muscle can continue to do the work without fatigue, without turn into lactic acid fermentation. Because without the oxygen they will turn anaerobic and lactate will build up in the muscle, it will cause the fatigue. So myoglobin tries to prevent that part and allow the physiological activities to continue in that very low level of partial pressure of oxygen. That's the job of myoglobin. It's a red colored protein that's found in our muscle, and this is much present in all the skeletal muscles that we know. It's very required in the skeletal muscles for proper functioning though. Okay? And another important thing, this oxygen dissociation curve are different for different person. It may be different for different person. For a smoker it's different, for a non-smoker it's different. For a person living in high altitude areas, their mold to that area, to that condition are more. So if you imagine, if you compare your oxygen dissociation curve with a Sherpa, what you will find out that the persons living on the top altitude, high altitude areas, they have a curve which is shift towards left, little bit like a myoglobin. So their hemoglobin is more eager, with a higher affinity binds with the oxygen, and they don't allow it to release the oxygen at a very less oxygen pressure, because they survive in lower oxygen pressure all the time. They live in lower oxygen partial pressure all the time. Okay, that's why it may be different for us to go to the very high altitude regions, but for them to live in that place and climbing the mountains become a lot easier. Okay, this part is really important. They have asked a lot of questions from this part in earlier exams, between the contrast of hemoglobin and myoglobin saturation curve, as well as between the individual saturation curve between human. For example, if you look at here, the oxygen saturation curve difference between an adult and an infant, and a fetal hemoglobin. If you look at a fetal hemoglobin, the curve is shift towards left. What does that signify? Again, it will remain attached with the oxygen for a long period of time. Why it's necessary? Because fetus is not going to get oxygen all the time. It takes some time for the oxygen to reach there, because the oxygen supply is transported by the blood from the mother. It took some time to reach there. So during that time period, they need to survive. So if there is any delay in oxygen supply from the mother to the fetus, They need to utilize this capability of holding on to the oxygen for long duration for survival. Okay, that's why this idea. So remember one thing, whenever you have this graph oxygen dissociation curve shifted to the right, it means affinity towards oxygen decreases, and if it's shifted to the left, affinity towards oxygen increases. Okay, so that's the part we end today. And if you should the topics that we discussed today, you read there, and especially for the other amino acid part, you need to remember a lot of things, and for this parts there are the concept that we discussed earlier. Okay, Haldane effect is the one when you look at the shifting of the curve to the left. Shifting of the curve to the left, and why it is required. Haldane effect is a portion where oxygen displaces carbon dioxide from the hemoglobin, and when it takes place, where it takes place, in the lungs, because in lungs carbon dioxide should be removed, released, oxygen should bind. And you know, if hemoglobin is too eagerly bind to the oxygen, then what will happen? It will never remove the oxygen, it will never release the oxygen. That is bad. And if the affinity of oxygen towards hemoglobin is very less, then it can remove the oxygen and it can never release the carbon dioxide. So these are these two problems. So in Haldane effect, what we can see in the lungs, the oxygen should displace carbon dioxide from hemoglobin. It should always displace the oxygen with the help of the carbon dioxide, and that is required to shift the curve to the left, because when it should happen, if the pH is high, if there is less metabolic wastes like DPG, and the temperature should be less. That's what the lungs all about. pH is higher in lungs compared to the other bodies or other parts of the tissue. Three-10 helix, I told you the idea of 310 helix. 310 helix, 1 and 1 plus C is a normal approach for the structure of 310 helix, and it is not genuinely as a alpha helix. It is a little thin, because you know, in alpha helix there are four, there is one extra amino acid to form and create a complete turn, but in 310 helix the turn is little thin, the diameter will be thinner. Okay, so let's keep it that way, and in the next class we'll be talking about the other part of the biochemistry. Okay, Thank you. Good night, everyone.